welcome everyone. Thank you for joining us today for our webinar on sequence alignment workflows in MegAlign Pro. My name is Stephanie Costello, and I'll be coordinating the webinar today. I'm here with Jackie Carville. She's on our marketing team, and she's going to be focusing on MegAlign Pro today. MegAlign Pro is one of our newest applications. She's going to go through several alignment workflows and different views in that module. You may have noticed that your phone has been muted. To ask a question, just type into the chat dialog and send the chat to the host. I'll then direct those questions to Jackie and will be answered for the whole group. And if you do need assistance during the presentation or the webinar, you can simply chat a message to me or email me at webinars@dnastar.com. And now I'll turn it over to Jackie.
Thanks a lot, Steph. So I'm just going to share my desktop here. And to get started, I'll be giving some background on DNASTAR as a company as well as MegAlign Pro itself. And then after the short presentation, I'll be getting into the software. So to get started, a little bit about us. We are a global software company headquartered here in Madison, Wisconsin. Our sales team, developers, and support network are all located here under one roof. So if you have questions, we have the full support staff to answer you, as well as use your feedback to make our software even better for the next release. Some history about us. We were born out of the E. coli lab of Dr. Fred Blattner, who was a professor of genetics at the University of Wisconsin in 1984. We built a strong reputation with our early products during the Sanger sequencing years that still continue on today. We still offer these classic sequence assembly and analysis tools. We have also expanded into the realms of next-gen sequencing, supporting all major next-gen sequencing platforms. In addition, we also now offer software to assist in the work of structural biologists. DNASTAR products have long been established on the desktop computer, but are now additionally available on the Linux cluster or on the new DNASTAR cloud. Now, DNASTAR is the leader in citations in peer-reviewed journals. For the past 28 years, more researchers have cited DNASTAR software in scientific journals than in any other sequence analysis software. Here at DNASTAR, we offer 3 suites for different areas of research. Now, over the summer, we had 3 separate releases with significant enhancements to these particular suites. The LaserGene Genomics suite provides all of the software necessary for next-gen sequence assembly and analysis. With the release of LaserGene 11, several new workflows in LaserGene Genomics suite were introduced. This release additionally introduced the DNASTAR cloud, as I mentioned earlier, making all of our software available up on the Amazon cloud. The Structural Biology suite provides tools for protein sequence and structure analysis. LaserGene 11.1, introduced over the summer, introduced our protein structure prediction program NovaFold to complement this structural biology suite. And finally, we have our Evolution suite, which we will be focusing on for the rest of today's presentation. With the release of LaserGene 11.2 in mid-September, new sequence alignment methods and tools were incorporated into the MegAlign Pro application within this Evolution suite. Now, this DNASTAR LaserGene Evolution Suite is our suite for evolutionary biologists to work with sequence alignments. This suite is designed around MegAlign Pro, the application we'll be working with today. Now, the applications in the Evolution Suite support a variety of workflows, including primer design, virtual cloning, gene discovery, Sanger sequence assembly, and, as we'll learn today, multiple sequence alignments. So today we'll be walking through 3 different alignment workflows in MegAlign Pro. First, we will perform a multi-sequence alignment. Second, we will go ahead and work with MultiSegment Files. And finally, we will perform a genomic alignment. So I'm going to get out of the presentation here and go into our software. The first thing I'm opening up here is the DNASTAR Navigator. Now, the Navigator is a guide to all of the DNASTAR applications. And in looking at the Navigator, you can view a summary of the DNASTAR applications that are available to you. Now, if we hover over different icons here, if I'm hovering over this MegAlign Pro icon, we can read a more in-depth description of each application and access additional resources such as videos, tutorials, and FAQs for that particular product. Now, the DNASTAR Navigator is also integrated with our help system to answer any of your questions. So it's very easy to search your particular questions about any of these applications and get those answered right within the Navigator. And I'm going to go ahead and open up MegAlign Pro from the Navigator here. And this opens up the MegAlign Pro interface. And for this first workflow, I'm going to spend some time introducing the various views within this MegAlign Pro interface, as well as walk you through how to perform a multiple alignment in MegAlign Pro. Now, in this workflow, we'll be adding protein sequences into our MegAlign Pro project and then aligning them using the cluster omega algorithm. Now, the first thing we need to do here is to add our sequences into the project. And we can do this in one of two ways. And the first is by selecting File, Add Sequences, or by selecting the Add Sequences to Project button from the toolbar here. So I'm going to go ahead and select this button from the toolbar and navigate to the data that I'm going to use for this particular project. And for this project, I'll be using 5 alcohol dehydrogenase protein sequences from different animal species. And so I'll highlight all these sequences here and select Open. And once I have these sequences opened in MegAlign Pro, we can take a look at the different windows in the MegAlign Pro interface here. Now, up at the top of the screen, we can see the Overview panel. And the overview provides a way to navigate within the sequences, no matter what type of sequences or alignment is used. Now, prior to alignment, as we can see here, each of our imported sequences appears as a green block with no gaps. And we can see that all of these green blocks have different lengths to them, which is proportional to the ungapped length of the corresponding sequence that we imported. Now, we can use this overview panel as a sequence navigation tool in a few different ways. This light blue region, as we see at the top of the panel here, is called the viewport, which shows the region currently displayed within the sequences view. Now, we can slide this viewport manually to scroll through the sequences and check out different areas here. Or we can simply double-click on any area or residue that we're interested in to center that viewport over that particular residue. Now, the sequences view below will display your added sequences, and after we perform the alignment, the aligned sequences. And now, if we look over to the right side of the screen here, we can see the Style panel, which offers us different options to customize parameters for various other views in MegAlign Pro. But before I go into more detail on the sequences view and the Style panel here, I'm going to go ahead and align our 5 alcohol dehydrogenase sequences. Now, as I mentioned, for this workflow, we'll be aligning using the cluster omega algorithm. And we can do this by selecting Align using cluster omega, or again, going to the toolbar and from the drop-down menu, selecting Align using cluster omega from this drop-down command. So after selecting that, the sequence has almost immediately aligned here. And if we now look at the overview again up here, we notice that the sequence blocks are now all the same length. We can also see sequence gaps within these blocks, which is represented by the gray coloring. Now that we have performed our alignment here, we can explore the sequences view a little more closely. Now, any of the sequences selected in this view are simultaneously selected in all of the other MegAlign Pro views. Now, at the top of the sequences view here, we can go ahead and see the ruler as well as the consensus sequence. Then in the sequences area below, we can see names and codes for each of our sequences. Now, the names of each of these sequences originated from the locus names that were embedded in our input files. Now, we want to be able to change these names so we can more easily perform our analysis on our alignment. And to do this is very easy. All we have to do is click on the sequence names in any of the views to select it, then right-click and choose Rename. And this pulls up this renaming dialog here. And I'm going to go ahead and rename the sequence file here. You can go ahead and do this for all of your sequence files to make this a little more easy for you to analyze in the future. So in addition, for any of these sequences here, we can expand the view by selecting this plus icon next to the sequence's name. And this will reveal a drop-down diagram with a details track containing features as well as a gap-aware ruler here. Now, if we wish to change the appearance of the sequences themselves, we can look to the Sequences bar within the Style panel, which we can see right over here in this section. Here we have the option to change the font style for any of these sequences, as well as the font size. And in addition, we can also have the option to select various different coloring schemes here, just to customize our projects to our particular specifications. So a final point about the sequences view here is that this view features the consensus match histogram down here at the bottom. And if we hover over a bar, we can see the percent of sequences that match the consensus at that particular residue. Now, this consensus match histogram is calculated by dividing the total score for the called consensus character by the number of sequences at that particular position. Now, as this agreement increases, we can see that the bar height also increases and becomes a lighter shade of green in this view. Now I'm going to go ahead and click on the Distance tab at the bottom, unless there's some questions that are coming in.
Yeah, Jackie, we had one question. Someone wanted to know how does someone know whether to use muscle versus cluster W?
Muscle versus cluster W. That all just depends on working with your own particular project. Since these alignments are really quick and easy to run, it's pretty simple to just run both of those alignments and then compare them to see what kind of results you would expect to get for that particular project.
Okay, thank you.
Awesome, thanks, Steph. Now I'm going to go ahead and click on the Distance panel here at the bottom of the screen, which we can access by this tab here. And this displays the distances view in MegAlign Pro. Now the distance view shows a matrix of numbers representing distances between each pair of sequences. Now by default, the distance view matrix is calculated using uncorrected pairwise distance. However, we can go ahead and alter these settings in the distance bar within the Style panel to use different distance metrics. And so we can see that this distance panel is right, or this distance bar is right here within the Style panel. We can go ahead and select any of these other various metrics available. Again, we can see that within the Style panel here, we have some additional customization options where, again, we can change the font if desired, as well as the font size, and even alter how many decimal places we would like to see displayed within our distance table here. So there are a few key tips that we should remember when interpreting the information shown here in the distance view. Now, first, larger numbers correspond to greater differences between sequences. And second, the upper and lower triangles of the distance matrix, as we can see here and here, are reflections of one another. So they will contain the same information. You shouldn't be surprised by that. Finally, values will differ for each of the various metrics used. As demonstrated earlier, if we switch between the different metrics, you'll see that these values will go ahead and change. And there are very useful resources actually within the DNASTAR help for assistance in interpreting this information for the specific metric you chose. And again, in response to the question, there would be some information located within the DNASTAR help, which gives some more specifics on these different alignment methods that you could read through to make the decision for your particular project. But there are a couple ways, since we're talking about the help, that you can have to access it. One is the help that's embedded within the DNASTAR programs itself, which I am opening up right here. So we can easily go through this help system and see more information on any of these views or workflows within the project. And to actually find the most updated help available, you can access the help from our DNASTAR website. All you have to do is, under the Support tab, select Online Help, and select the help for any of the applications that you're interested in here. So I'm going to go ahead and go back to our project in MegAlign Pro. And as one final note on this distance matrix here, you can easily export this distance matrix by selecting File, Export, Distance Matrix here. And then you can go ahead and save that wherever you'd like. The last view we're going to examine for this particular project is the tree view, which we can then access from this tree tab here at the bottom of the screen. So I'm going to go ahead and select that, and we can see our nice tree that was created from this particular project. Now the tree view is calculated by using whatever distance matrix I had, or metric that I had selected. And again, you can change this distance metric in the style panel as we did earlier. As we change these distance metrics, these are synchronized between all views. We don't have to do this twice for both the tree view and the distance view. You can also use the horizontal and the vertical zooms on the side of this to adjust your tree for easier viewing options. And finally, in the style panel, as with all the other views, we do have options to adjust the layout of our tree as desired, to go ahead and change the font on the leaves and the size as with the other views. And then we can even go ahead and change the color and even the width of these different branches to make them customized for our particular project. Now, this wraps up our workflow on performing a multiple alignment in MegAlign Pro. So now I'm going to go ahead and show you how to go ahead and work with multi-segment files in our next workflow here today. So I'm going to go ahead and from the toolbar here, open up a new alignment project. And as a little background on this second workflow here, sequences that consist of more than one chromosome, contig, or fragment are called multi-segment files. So let's say that you want to string together the consensus sequences from all the contigs in an alignment, or chromosomes in a genome, or exons in a gene. Now, if you simply added these sequences as we did in the previous workflow, you'll end up aligning these parts to one another. Now, instead, we need to enter in the sequences using a different method to make sure that the entire set of sequences in the multi-segment file will instead be treated as an end-to-end concatenation. So instead of selecting File, Add Sequences, as we did with the last workflow, we're going to instead select Add Sequences End-to-End. Additionally, if I click Cancel here, you can also find this Add Sequences End-to-End option right here in the toolbar. So you can access this dialog window in these two different ways. So I'm going to go ahead and click on the Add button here and navigate to my data. And for this particular project, we're going to be using nucleotide files that represent the RP1 exons for 7 different mammal species. Now, each exon here consists of 3 to 4 different segments. So the first thing that we're going to want to do here, if I expand this window up a little bit, is to sort all these files by name, because we really want to ensure that the sequence files are in alphabetical and numerical order for these particular species. And so after I've done this sorting, I want to click on these 4 chimp sequences here and select Open. And I'm going to go ahead and rename this group name as chimp. And before I move on to add my next set of sequences, I'll want to select the Add Another checkmark tab so that we can add these in succession here. So I'll click OK. And we can see that this first sequence has already been added into our MegAlign Pro project. And I'm going to go ahead and do this for our second group here, which I'll rename here as cow. Now I'm going to want to do this for this project for several different groups. So in addition to chimp and cow, I also want to create these sequence groups for dog, human, mouse, and rat. But so I spare you watching me import all of these sequence groups. I'll just go ahead and import the last sequence group of interest here. And we'll view the completed project in our next window. So I'm going to want to input these rhesus sequence files here. I'm going to go ahead and add these sequences. And as you may have noted earlier, we have made sure to create our groups specifically with the sequences in order. So for this last group that we have to create, I want to make sure and deliberately order these sequences incorrectly so that we can see what happens to the project when we do this. So instead of ordering them 1, 2, 3, 4, I've done this 4, 2, 3, 1 here. And select, and I'm going to, since this is the last group I want to add, I'm going to remove the Add Another checkbox and select OK. We can see that all of these were added here. But just since I want to move forward here with the presentation, I'm going to go ahead and go to a project that's already been aligned with all of these sequence groups created. And for this particular project, we were aligning with the muscle algorithm. And again, we can do this by selecting Align using muscle, or under this drop-down tab here, selecting Align using muscle from the toolbar. But so I'm going to go ahead and open up this sequence project here, which as you can see in the sequences view, we've now added all of the sequences groups. And where we can see by the same length of all of these green sequences that this project has already been aligned here. So if we look again at the overview window here, the first thing we want to look for in our particular project is both the longest segment as well as the longest gap region in each of these sequences. Now for most of these groups here, we can see that this longest segment is off here to the right, and that this longest gap region here is, again, at the end of this longest rightmost segment. However, for the rhesus group, we can see that both of these, both the longest segment and the longest gap region, are located here on the left. So if we remember, we did deliberately add these sequences in the wrong order. So when using your own sequences, this is usually a pretty good clue that the sequences you imported were added in the wrong order, as we did here. But luckily, it's very easy in MegAlign Pro to remove a group and then simply add it again in the proper order. So all we have to do is right-click on this rhesus group and select Remove Sequence. Select OK. And again, we can go back to our Add Sequences End-to-End in Project. Name this as our rhesus group. Go ahead and add these 4 sequences here, making sure they're in the right order, and select OK. And when we do that, this dialog here appears. And we can go ahead and simply click Align to perform our muscle alignment again, but this time with the rhesus group in the correct order. Additionally, if we decided after doing this alignment that we really didn't want to align with muscle, we wanted to align with cluster omega or mauve, we could select this Align with Options button to select a different alignment method and adjust the parameters for that particular method. And in addition to that, if we decided that, maybe we didn't want to align these sequences in the first place, we can go ahead and choose to unalign all of these sequences. This particular alignment takes a couple minutes to complete. So instead of realigning this project, I'm simply going to open up another project that I've already performed this alignment in. Now once this alignment has finished here, we can go back and again look at the overview segment. And now we can note that, if I expand this view down a little bit, that this largest gap region in the overview has now disappeared. And we can see that these longest segments here are all already matched up in the correct order here. And there are only small gap regions present in this project. So now for this particular alignment, I'm going to go ahead and examine the tree view. And what's important to keep in mind when looking at the tree view here is that this tree view is a phylogenetic tree that graphically illustrates plausible evolutionary relationships for these 7 species. And this will all change based on the metric used. So if we look at the tree now, and we go over to the Style panel, and we change this from this uncorrected pairwise distance, we can see that the groups dramatically then will change within the tree. One last interesting view to take note of in this workflow is the console view. And we can go ahead and select View Console. I'm going to expand this up so we can take a closer look at it. To take a look at this view. What we just did here is we opened up a log that shows the progress of your alignment. And for an alignment in progress, the console view displays the start time and progress information. And for a completed alignment, such as this one, the console displays statistics for the alignment as well as start and end times. So this wraps up our workflow for multi-segment files.
Yeah, someone wanted to know if there's a limit to the number of sequences or genomes they could align and the length of them.
Sure, yeah, they're really, MegAlign Pro here is memory limited, not number limited. So really, depending on how much memory your computer has, you're pretty much unlimited by your sequences. I can't give you like a straight number on that, but it can handle very, very large amounts of sequence files here.
Okay, thank you.
Great, thanks, Steph. I'm going to go ahead and close this console tab here. And now I'm going to go ahead and demonstrate our final workflow, which is performing a genomic alignment. And for this particular tutorial, we're going to be adding 6 nucleotide sequences to MegAlign Pro and then aligning them using the progressive MAUV algorithm. And as a little background on this, the MAUV algorithm is specifically designed to align genomes, which is a workflow that's usually impossible or at least extremely difficult with other alignment methods. Now, there are a few characteristics of these whole genome alignments that cause problems in these other methods. The first thing here is that these whole genomes have a large memory requirement. Second, genomes can be very divergent. Genomes can have a lot of rearrangements, including both large and small insertions, deletions, duplications, and inversions. Some genomes may also have gene clusters or large islands of unique sequences. And finally, you may also run into some problems with transposition in eukaryotes, where entire sections of chromosomes in one genome may be transposed to an entirely different chromosome in other genomes. Now, the MAUV alignment method can, to some extent, surmount all of these issues. How this method works is to break the problem into smaller alignments, and then at the end, merge as many of these small alignments together as possible. So we'll go ahead and get started here with our MAUV alignment. And what I quickly wanted to point out here is that there is one more way to add sequences to MegAlign Pro, as well as to any of our other laser gene applications, and that's to simply drag and drop your sequences into the project. So if I opened up my Explorer window here, I could really easily just select all of these sequences and drag them into any white space here and set up my project that way. Since these sequences are very large, you can see it does take a few minutes to entirely load all of these sequences. So there we go. They all just popped up right here. That went pretty quickly. So I have all these sequences here ordered in the way that I want them. And we can see that we have all of these sequences unaligned here in the overview window. So for the previous 2 workflows, we've used the clustral omega and muscle alignment methods, and I've also used their default parameters by simply selecting align using muscle, or whatever method we used, or selecting it from this drop-down toolbar. But for this tutorial, since we're going to use MAUV, we'll also explore how to alter parameters prior to alignment. Now, for most alignment projects here, the default settings are entirely suitable, including this one. So we could very easily just select align using MAUV, and we would be fine here. But for this particular workflow, what I'm going to want to do is adjust MAUV's seed weight parameter here. So what I'm going to do is choose align with options, and this will pull up the alignment options dialog. And if we select between the different alignment methods we have available, we can see that for each alignment method, we do have a wide variety of different options that we can choose to alter for our particular alignment. So I've selected MAUV from the alignment method here in the alignment options window. And I'm going to go ahead and lower our seed weight to 13 here. So when aligning divergent genomes, lower seed weights may provide better sensitivity. And seed weight is the minimum weight of the seed pattern used during the first pass of a multiple alignment. However, it's important to remember that if we set this value too low, the result will be reduced sensitivity. So that's why I only reduced the seed weight here from 15 to 13. And we can now go ahead and click Align to perform the alignment. Now this does take several minutes to align, just since we are working with whole genomes, but really this alignment can only should take about 5 to 10 minutes. So I've already performed this alignment, so we don't have to wait during the webinar here. So I'm going to go ahead and look at this. And in looking at this completed project here, we can see that MegAlign Pro displays the genomes as locally collinear blocks, as opposed to the green blocks that we saw with the other alignment methods. Now these blocks here represent multiply aligned sections of at least 2 of the current set of genomic sequences. Discontinuity in numbering is displayed as a break between the different blocks. And connecting lines, these black connecting lines that we see, will show the correspondence between the blocks. Now if you notice blocks hanging below the black line, as we can see in these first couple sequences up here, this indicates that the hanging blocks are on the opposite strand compared to the other blocks. And we can also see that all of these blocks are of different colors. And if I double click on a block, let's say I'm interested in this gold block here, we can see that the gold blocks are now in a single blue highlighted column. MegAlign Pro will also flip strands if necessary to orient properly for the new alignment. We can see it do that if we click on the pink blocks here. You can see it flip those strands pretty quickly. Once we select these certain blocks, the viewport is then located in the leftmost end of those blocks. I'm going to go ahead and zoom in a little bit on this mauve alignment so we can see this a little better. And I'm doing this by using these vertical zoom sliders as well as the horizontal zoom sliders to adjust our views. Now in the sequences view down below here, we can see that the residues outside of the selected box are shown in a dim version of the color scheme. And these represent flaking sequences immediately adjacent to the currently selected block. And these sequences appear as unaligned, ungapped, and without a consensus. If we want to see additional details for these dimmed sequences, we're going to have to select the corresponding block in the overview. In addition, the gap histograms aren't shown in this view. They're only shown for the currently selected block here. Now if I go to the distance view, or the tree view, now these views are specific only to our selected block here. So if I select this blue colored block, we can see that some of these numbers have gone ahead and changed in this distance view here. Sorry about that. So if we'd like to order the sequences in all the other views the way they appear in the tree, we can also simply select Tree, Order Sequences Like Tree, and we can also have the option to reorder our sequences that way. Now for the alignments conducted in MegAlign Pro, you do have a variety of exporting options available to you. First, the aligned sequences can be exported as FASTA files or as MSF files. And in addition, you can also export phylogenetic tree or distance matrix for your alignment. And as we saw before, any of these export options are very easy to do right here from the file menu here. So we can export our selected alignment block. This is what we can export as a FASTA or an MSF file. And then additionally, we can export the distance matrix or tree and save those files to our computer as well. In addition, for all the long-term LaserGene users who may be listening today, you can have a one-click export option right here to send any of these MegAlign Pro alignments over to MegAlign. Our MegAlign application also houses some pairwise and multiple sequence alignment algorithms separate from MegAlign Pro, including other Clustall algorithms, Jotunheim, Wilbur-Lippmann, Martinez-Needleman-Wunsch, Littman-Pearson, and dot plot analysis, to name those. In addition, in MegAlign, you do have some bootstrapping capabilities that are available within this application. So if you wish to perform some bootstrapping analysis on your project, that's a good method to do so. Now that wraps up the 3 workflows I'd like to present here today. Just to recap, we've gone ahead and looked at a multiple sequence alignment workflow using Clustal Omega, workflow using muscle and multi-segment files, and finally a genomic alignment using MAUV. Thanks again for joining us today. I'd be happy to answer any questions.
Yeah, we had a question. Someone wanted to know how our MAUV that we implemented compares to the MAUV that someone can just download on their own.
Sure. So the MAUV that you can download on your own isn't going to have this nice interface here where you can examine these locally collinear blocks. It's strictly a command line program, so you don't get that alignment housed within this interface for various styling options as well as other analysis options here.
Okay, great. We did have a few more technical questions we didn't answer. We will have someone from tech support personally respond to you after the webinar and send you an email or give you a call. If you think of other questions later, you can always email those to webinars@dnastar.com. I also wanted to point out that on our website, looks like Jackie's going there now, under the support tab, we have a full videos page that has lots of different videos that you can look for any of our applications. We also have a webinar page on our website as well that will post upcoming webinars. So please check that for ones that you might be interested in and register for those. Again, if you have any questions, send those to us directly. And thanks everyone for joining us today.
Thank you.